How do cells communicate so efficiently to coordinate the complexities of life? While chemical signaling through hormones and neurotransmitters is well established, recent discoveries suggest that cells might also use other methods, such as electromagnetic fields, mechanical signals, and even light, forming what could be called an intercellular internet. This idea proposes that cells might have fast, non-contact communication systems that go beyond the slower diffusion-based chemical pathways. For example, electromagnetic signals generated by moving ions might allow cells to synchronize activities over larger distances, much like a wireless network. Similarly, biophotons, weak light emissions from cells, could transmit information at incredible speeds, coordinating processes like repair and growth. These unconventional pathways open new frontiers in understanding health and disease. They could explain phenomena such as the rapid spread of cancer signals or how neurons influence each other beyond synaptic transmission. If we can harness these non-chemical mechanisms, it might revolutionize medicine, offering treatments that target electromagnetic signals in tumors or enhance biophoton-driven healing. However, exploring this hidden layer of communication is challenging, requiring advanced tools to measure subtle signals and integrate them into existing models. Unlocking this intercellular internet could reveal entirely new ways in which life operates.
Cells within an organism constantly exchange information to coordinate functions, maintain homeostasis, and respond to external stimuli. Traditionally, this communication is understood to occur through biochemical signaling, such as the release and reception of hormones, neurotransmitters, and cytokines. However, emerging evidence suggests that cells may also utilize physical and electromagnetic mechanisms, hinting at a more complex intercellular internet that goes beyond conventional chemical pathways. This paradigm could reshape our understanding of cellular networks and their role in health and disease. Example: neurons communicate through synaptic transmission, where chemical neurotransmitters cross synapses, but recent studies suggest that electromagnetic fields might also modulate neuronal interactions. Analogy: cellular communication through chemical signals is like traditional mail delivery, effective but limited in speed and scope. The hypothesized intercellular internet, in contrast, resembles a high-speed wireless network. Chemical signaling has inherent constraints, including its reliance on diffusion, specificity, and molecular degradation. Diffusion is often slow and limited by distance, making it less effective for rapid or large-scale communication. Additionally, chemical signaling requires the synthesis and secretion of signaling molecules, which can be resource-intensive for cells. These limitations raise questions about how cells coordinate complex processes, particularly in dynamic environments where speed and adaptability are critical. Example: the immune system relies on cytokines to signal the presence of pathogens, but the slow diffusion of these molecules can delay the response to rapidly spreading infections. Analogy: chemical signaling is like sending messages by courier, reliable but often too slow for urgent or widespread communication needs. Cells generate and respond to electrical signals, particularly in the nervous system, where action potentials propagate along neurons to facilitate rapid communication. Beyond neurons, other cell types, including cardiac and epithelial cells, exhibit electrical signaling through ion channels and gap junctions. These signals provide a faster mode of communication than chemical diffusion, allowing for the synchronization of cellular activities. Example: cardiac cells synchronize their contractions through electrical signals transmitted via intercalated discs, ensuring the heart beats in a coordinated manner. Analogy: electrical communication is like a wired telephone network, faster and more direct than traditional mail, but still constrained by physical connections. Mechanical forces and vibrations are another potential medium for cellular communication. Cells sense and respond to mechanical cues from their environment through mechanotransduction, converting physical stimuli into biochemical signals. Mechanical waves, such as vibrations within tissues, could serve as a rapid and energy-efficient means of signaling, particularly in densely packed cellular environments. Example: bone cells, osteocytes, communicate mechanical stress to osteoblasts and osteoclasts. Coordinating bone remodeling in response to physical activity. Analogy: mechanical signaling is like Morse code transmitted through vibrations, simple and efficient for specific, localized messages. Extracellular vesicles EVs, such as exosomes and tunneling nanotubes TNTs, represent emerging mechanisms for cellular communication. EVs carry cargo like proteins, lipids, and RNA between cells, functioning as molecular messengers. TNTs, on the other hand, are physical connections that allow the direct exchange of organelles, ions, and even electrical signals between distant cells. These structures highlight the diversity and complexity of intercellular interactions. Example: cancer cells release EVs containing RNA that can reprogram nearby cells to support tumor growth, demonstrating how EVs contribute to pathological signaling. Analogy: EVs are like courier packages, delivering specific payloads, while TNTs resemble private pipelines connecting distant locations. Recent research suggests that cells might communicate using electromagnetic (EM) fields generated by the movement of charged ions and molecules. These fields could provide a non-chemical, non-contact method for coordinating cellular activities across larger distances. EM-based communication might explain phenomena like synchronization of cellular activities in tissues and the influence of external EM fields on biological processes. Example: studies show that cells exposed to specific electromagnetic frequencies exhibit changes in gene expression and behavior, suggesting they can detect and respond to EM signals. Analogy: electromagnetic communication is like a Wi-Fi network, enabling fast, wireless transmission of information across large areas. Biophotons, weak emissions of light by living cells, represent another intriguing avenue for intercellular communication. Generated during metabolic reactions, biophotons might act as signals, transmitting information at the speed of light. This hypothesis is supported by evidence that cells can detect and respond to these emissions, potentially coordinating processes like growth, repair, and immune responses. Example: in plants, biophotons emitted by stressed cells have been observed to influence neighboring cells, suggesting a light-based signaling mechanism. Analogy: biophotons are like fiber optic communication, using light to transmit information rapidly and precisely. Understanding these non-chemical communication mechanisms could revolutionize medicine. Diseases like cancer, neurodegeneration, and autoimmune disorders often involve dysregulated cellular communication. Targeting or modulating these alternative pathways might offer new therapeutic strategies. For instance, disrupting the electromagnetic signaling of cancer cells or enhancing biophoton-mediated repair mechanisms could lead to innovative treatments. Example: altering the electromagnetic environment of tumor cells using specific frequencies has shown promise in inhibiting their growth in experimental models. Analogy: targeting non-chemical communication is like hacking into an encrypted network to disrupt harmful messages or restore beneficial signals. While the evidence for non-chemical cellular communication is growing, significant challenges remain. Measuring and manipulating subtle signals like electromagnetic fields or biophotons requires advanced technologies and interdisciplinary approaches. Future research should aim to clarify the mechanisms, specificity, and biological significance of these pathways. Understanding how they integrate with traditional chemical signaling will be critical for developing a unified model of cellular communication. Example: advanced imaging techniques such as quantum-based sensors are being developed to detect weak electromagnetic fields and biophoton emissions in real time, paving the way for new discoveries. Analogy: investigating intercellular communication beyond chemical signals is like decoding a hidden layer of a complex operating system. Unlocking these secrets could reveal entirely new functionalities.